Hi, my name is Maarten Verhoeven and today I would like to tell you more about our call to partners for the TKI project Pathfinder. Within this project, we have the ambition to take big steps in understanding flavor formation during fermentation on plant-based substrates. Let me first introduce us. For this project, people um, from Wageningen Plant Research and Wageningen Food and Biobased Research joined forces to provide unprecedented level of expertise not only on fermentation but also plant-based substrates on the molecular level. Together we conceptualized the Pathfinder project, and together combining world-renowned expertise on plant-based substrates and food fermentation, we hope to really make a next step into this fascinating field of food fermentation. Fermentation promises huge potential for food ingredients to provide complex flavors for food, for things like plant-based meat or dairy or fish alternatives. However, we have observed that this is not trivial. It is not trivial to yield a desirable flavor palette. When it comes to flavor, we as consumers are very sensitive when it comes to changes in flavor composition. We are, however, not starting from scratch. Work has been performed at WFBR, have shed significant light on how fermentation in multitude of microbe and substrate combinations have affected formation of flavor palettes. With fermentation, you often see that um, individual flavors are either absent or under or overrepresented. And that is an important key part here, because if yielding a flavor composition that fully resembles meat, dairy, or fish is therefore remains a challenge. Additionally, what you also see is that plant-based substrates often introduce off flavors as well. Uh, in this domain, we are currently lacking, as a scientific field, a lot of data, methods, and know how to mitigate this. Within Pathfinder, we are trying to find a solution by tailored fit of substrates and microbial metabolic pathways to yield the desired flavor palette. So this will require a holistic approach that looks at all three aspects. So we're looking at both these plant-based substrates on the left, the microbial conversion, and really zoom into the microbial pathways that are part of that, significant part of that conversion, and obviously the fermented food, the product at the end. But also we have to look at how to characterize the end product that we are aiming for. That can be a meat, dairy, or a fish-based alternative, but it can be any other kind of food application in which fermentation could play a big role. Pinpointing the gaps in flavor chemistry is then that can be filled with fermentation is then one of the main goals we want to achieve. So really focus on where microbial conversion can have a big impact, and that will generate a data library of relevant flavor conversion pathways. Really looking at the metabolic conversions within the cell and see if we can link that towards the particular flavor generation during fermentation. Um, Additionally, we would like to identify substrates that can be used as flavor precursor supplements. So prior to a ferment, we could add a certain flavor precursor for certain flavors that are for certain metabolites that are lacking within the plant-based substrates we are looking at. So for that, it is important that we generate a big library of plant-based substrates and microbial strains that we map together to finally end a certain overall data set. Alternatively, we could also look at how we could supplement flavor after fermentation. So if we see that a certain fermentation is lacking a certain flavor, we could add this afterwards and see if that would be cost effective. And finally, you also often see that plant-based substrates introduce off flavors. And this is also, to us, an underexplored field in which we look at whether microbial conversions can help to mitigate that and remove those flavors. For all this, um, a good understanding of what these microbial conversions entail is key. So one of the major activities will be to look at in silico study in which we take the genomic data from microbial strains. That can be strains that you bring in as a partner, but could also be key representatives of species that we obtain from publicly available libraries, and use that data to understand better how these conversions work. And also give predictions on whether we think one or the other species can be of relevance for the conversion of a certain or the production of certain flavors. Additionally, we really need to characterize biomass to a very specific, very high detail. This is also where plant research comes in. They have really a lot of expertise in what kind of metabolites and flavors and molecules are present within plant-based substrates. And here also US partners can be of really great help by introducing us with relevant substrates and also all the data and expertise you have on that part. The final end goal is to see if we can source plant-based substrates that can serve as a supplement to add flavor precursors to a fermentation. So imagine here a scenario which you would have a high protein plant-based substrate such as faba or a soy-based substrate, and we would add another plant-based substrate that is rich in flavor precursors on top of that to enhance the fermentation. You could think of things like cauliflower, which is high in sulfur compounds, which helps to maybe induce the formation of flavors during the fermentation. So here an overview of the potential work package structure. We would like to divide this specifically at the beginning in two different kind of work streams that contain different work packages. Characterization is key. We really need to have a good understanding of what we are working with. So we already have quite some elaborate GC and LC MS methods to test for the presence of 60 key flavor compounds. But for this project, we need to bring this to a level that we can really quantitatively rely on this data. So that's why this is an important part. You can benefit from that as well as partners by bringing in substrates or end products of your own, and we can evaluate this in the same setup. Then we have the pathway and precursor identification. Here we have a targeted and an untargeted approach. In a sense, the targeted approach is built upon the knowledge we will generate within in silico genome analyses of these key representative species and the species you bring in, and then use that data to really pinpoint the potential pathway precursors and bioconversions that are related. Together with the untargeted approach, this will also gain a lot of additional knowledge on how to really apply this in the lab. So when you look at the untargeted approach, here we will actually start by screening in all kinds of high throughput screening methods we have already, which we can screen thousands of different ferments and fermentations and measure the pH and also control pH during the fermentation. But on top of that, we can also use GC and LC MS methods to really get a good sense of what kind of flavors are being produced and what kind of off flavors are present, you name it. All that information will give us a lot of insight on what is, so to say, possible with fermentation with different combinations. And then with that, we want to go a step further and really go into looking whether we can use combinations of substrates and co-substrates and specific flavors and flavor precursors to make tailor-made fermentation possible. That will be the final part of the work package overview. So, how can you participate? Well, I really think that together we can bring utilization of substrates, flavor compounds, flavor precursors, microbial strains for the production of fermented food intermediates to really a next level. Combining our efforts really helps us to uh, produce this new workflow for tailored fit fermentation of substrates, co-substrates, metabolic pathways, and the accompanying strains that are underlying. Uh, for you as a partner, the characterization by itself of your brought-in strains, but also substrates, can be of great value. And I think that is a main benefit of a combined project as this one, where for all partners can benefit equally from the facilities that we have and the expertise we have at WFPR, and also the knowledge that is brought in from all parties to bring this to another level. So this characterization is screening on relevant substrates in this high throughput manner, and also when it comes to the genomic-based pathway analysis, we will take along at least species representatives from public libraries to really give you an insight on what we think are the microbial conversions behind it, and to combined, we hope we can really bring it forward. On top of that, we could also look at key existing products. So, if you're interested in generating a plant-based version of an existing product, this could be an opportunity to dive into what is the flavors that we need to generate there, and how can fermentation help there. So, with that, I would like to thank you very much for your attention. We really hope that a lot of you will come back to us and bring a lot of interest, and we will really look forward to your responses. Thank you.